Recording in progress.
A very good afternoon to everybody. First of all, I request everybody to keep your mobile in silent mode. So today we are here for the international webinar on citrus horizons, genomic tales, germplasm utilization and cultivar chronicles. Before proceeding to the program, I'll request Nikhil to, we'll start the program with ICAR Geet.
जय जय कृषि परिषद भारत की सुखद प्रतीक हरित भारत की कृषि धन पशुधन मानव जीवन दुग्ध मत्स्य फल यंत्र सुवर्धन वैज्ञानिक विधि नव तकनीके पारिस्थितिकी का संरक्षण शस्य श्यामला छवि भारत की जय जय कृषि परिषद भारत की। हिम प्रदेश से सागर तट तक मरुधरती से पूर्वोत्तर तक हर पथ पर है मित्र कृषक की शिक्षा शोध प्रसार सकल तक आशा स्वावलंबित भारत की जय जय कृषि परिषद भारत की जय जय कृषि परिषद भारत की।
So once again, a very good afternoon to everybody. So today, on this occasion, we have 4 foreign delegates among us. Dr. Fred Jimiter Jr., Professor Citrus Breeding and Genetics and Genomics at UF Citrus Research Education Center. And Dr. David A. Karp, Assistant Specialist, Department of Botany and Plant Sciences, University of California, Riverside. Dr. Chandrika Ramadugu, Project Scientist, University of California, Riverside, USA. Dr. Georgios Viadalakis, Professor Plant Pathology, Director, CCPP. So first I would like to request our director to welcome our guest with a bouquet. So first I'll request the bouquet to Dr. Fred Jimiter Jr. Now I request to Dr. David Karp. Now I request to provide bouquet to Dr. Chandrika Ramadugu. And to Dr. Georgios Vidalakis. Now I request Dr. Vijay Kumari Madam to invite our director with a bouquet of flowers. Now I request our director for his welcome remarks.
Good afternoon, everyone. I first of all welcome all of you, especially the dignitaries on the dais, esteemed Professor Fred Jimiter, who is a professor in citrus breeding, genetics and genomics at University of Florida Citrus Research and Education Center, Lake Alfred, USA. His research has and then Professor George Vidalikis from University of California, then Dr. David Karp, again from University of California, and then Dr. Chandrika Ramadugu, and my colleagues, scientist colleagues, who are present here, and other distinguished my colleagues, citrus workers, who are attending this program, international webinar by virtual mode, from different institutes, state agriculture universities within India and outside as well. Let me first introduce Professor Fred Grimiter, who is a professor in citrus breeding and genetics and genomic at University of Florida Citrus Research and Education Center. His research has included studies into the genetic control of commercially important traits such as fruit quality and disease resistance, particularly against citrus greening disease, that is HLB. Understanding the host pathogen interactions in HLB, then developing genomic tools for selection in breeding and its possible use in modification of different commercial citrus cultivars to develop tolerance or resistance against HLB. His team at CREC has released more than 30 new citrus rootstocks and scions. Personally, I had the opportunity to do few experiments in his lab while I was working as a visiting scientist at CREC during 2010 and again during 2000. Professor Grimiter previously, he led the International Citrus Genome Consortium, and his team first time published full genome sequence of citrus in 2011. He had several publications in Nature Biotechnology, Nature Communications, and Nature that have focused on origins, evolution, and domestication of citrus species as revealed by his extensive work on comparative genomic studies. I welcome you, Professor Grimiter, to our institute, that is ICAR, Central Citrus Research Institute. I also welcome Professor George Vidalicus, a professor of plant pathology and my old friend, who is an extension specialist in plant pathology in the Department of Microbiology and Plant Pathology at University of California, Riverside, where he serves as the director of Citrus Clonal Protection Program. I had the opportunity to see the extensive work being done by Professor Georgius during June 2023, very recently, where I visited along with the delegates from Asian Development Banks. George is working directly with the citrus industry, the growers, the citrus nursery owners at California State, as well as national level in United States, in a series of different citrus pathology and regulatory issues. And his team has developed a research and extension program that is specifically focused on the study of all major graft transmissible pathogens, virus, viroid, bacteria, their diagnostics, their therapy, production and distribution of pathogen-tested citrus germplasms, and propagative material. I welcome you, Professor George, in our institute in this afternoon. I also welcome Dr. David Kerr, who is working as assistant specialist in the Department of Botany and Plant Sciences, University of California, Riverside. His team did research on different citrus variety collections with a major focus on citron that has been collected from around the globe for the last more than 20 years. Dr. David and his team has recently published Citron Compendium that has been published by Swinger in 2023. He has also compiled the modern citrus cultivars descriptive database, which describes a large number, it's more than 1,200 citrus cultivars from around the world. I welcome you, Dr. David. And finally, also, I welcome Dr. Chandrika Ramadogu, is a project scientist at Department of Botany and Plant Sciences in University of California, Riverside. She is working on citrus genetic diversity and disease resistance aspect. And her team are engaged in genome-assisted breeding to integrate citrus HLB resistant traits from Australian limes, which are resistant to HLB, into cultivated citrus. Lot of good results Dr. Chandrika have obtained and her team that she recently explained in our recently held Asian Citrus Congress that we organized in Nagpur during October 28 to 30th October here in Nagpur. I'm very sure the research work being done by Dr. Chandrika and her team will certainly benefit, not only benefit the multi-billion dollar citrus industry of California, Florida, or Texas in US, but also other countries of the world, including India. I welcome you, Dr. Chandrika. I see several hundreds of people, citrus workers, not only in this place, but also those who have joined through online mode from different other institutes in the country, and few are also from outside the country. And I welcome all of you in today's international webinar on citrus origins, genomic tales, germplasm utilization, and cultivar chronicles. And I all of you to listen very carefully that today's very, very important 4 lectures being delivered by the World Authority in this aspect, all the 4 citrus experts, those who have come from USA. There will be special interaction session discussions once the 4 lectures are completed. Without taking much time, once again I welcome all of you and in this program, and thank you very much.
So now I request everybody to kindly seated below so we can start the presentations. So we'll start with our first presentation titled Comparative Genomic Analysis Illuminates the Evolution and Domestication of Citrus, the Next Chapter. So this is delivered by Dr. Fred Gemeter Jr. He is Professor Citrus Breeding Genetics and Genomics at UF Citrus Research and Education Center in Lake Alfred, Florida, USA. So we will having the discussion at the end of all the 4 presentations. So first we will have all the 4 presentations in trot, then we will have the discussion. So now I request Dr. Fred Gemeter to start with this.
Shall we begin? Good afternoon, everyone. I'm very happy to be here in Nagpur, my very first visit to Nagpur. I've known about this center for a long time. I met Dr. Ghosh many years ago now, when we both had a little bit more hair and were a little more trim and fit. I also know my friend here, Ashish Das, for quite some time as well. He also worked in my laboratory, and we've had some memorable visits in different places together over the years. So it's good to be here. I'm going to tell a story today. It's not my story. It's a story that's been written by many different people together. It's been an international collaboration. I'm speaking on behalf of a large group of researchers who've worked together to do this. And the story has to do with the origins and the evolution of citrus, and how citrus genomic science has helped us to understand that process. Many ideas have been out there over the decades about the relationships of different kinds of citrus varieties, about what is a species, what is not a species, and so on. But with the advent of genome sequencing and comparing these genome sequences, we've been able to answer many of the questions that have existed for decades. Back one, please. So there is a picture of the tallest mountain in the world, Mount Everest, the Himalayas. And in the foreground, you can see images of our favorite plants in the world, citrus plants. And what is the connection of citrus plants to the Himalayas? The role of geology and plate tectonics has been critically important for the diversification of citrus species. And we've traveled in many of the places where citrus is native, particularly in China and other parts of Southeast Asia. This was my first opportunity to visit Northeast India, which is another part of the story, a critical, important part of the puzzle. Okay, next slide. So as we look at a photograph of the morphological diversity of citrus fruit, we see many different sizes, shapes, colors. In your mind, I think you can imagine the smell and the flavor as you look at these different things. Citrus is something that's important to all humanity, and we all are wired to citrus fruit. But there's a lot of diversity that we can see. And on this slide, it illustrates some ways that people in the past have categorized these diverse types. They've looked at the nature of the leaves or the nature of the fruit. Are the fruit large or are they small? Do they have many locules or few locules? Are the flowers compound or very simple? And so on. These are all morphological characteristics that historically have been used to define these different kinds of citrus groups. But many times these sorts of measures are more subjective than they are objective. And they can be influenced by environment. They can be influenced by human opinions. So we have morphological characters. People have used chemical characteristics of the plants as well. They've looked at different volatile aromatics, polymethoxylated flavanones, peel oil volatiles, and use this kind of information to define what are species and look at the relationships. But with genome sequencing, now we have data that are not influenced by environment, not influenced by human opinion. They are data that are real and immutable. So it's a very solid basis for us upon which to base our decisions, our ideas, our conclusions about the relationships of different kinds of species. Next, please. In the late 2000s, a group of us internationally came together in an effort to sequence what we called at that time the citrus genome. And we looked at trying to find $16 million among our different countries, our labs, our organizations to invest together. This was right at the time when the first human genome sequence was published, and we knew it would take millions of dollars. Today, we can sequence dozens of genomes in a week for just a few hundred US dollars. The challenge in interpreting it is much greater, but the technology has changed. Nonetheless, at that time, we began to look at 8 different kinds of citrus. We had 2 pomelos. This doesn't really show. Two pomelos representing Citrus maxima, sometimes called Citrus grandis. We had Ponkan mandarin, which you know as Nagpur Santara, same thing. Our assumption was that this was Citrus reticulata. This was the representative. In fact, the type specimen of Citrus reticulata is just this cultivar, although they called it Batangas when they found it in the Philippines. But this was defined as Citrus reticulata, the mandarin. We had another mandarin that we also believed to be pure reticulata called willow leaf. We were very much interested in sweet orange because everywhere in the world citrus is grown, there's always some sweet orange grown, regardless of the industry. We looked at sour orange and a few other mandarins that we wanted to study. So when we look at genome sequence, one of the tools that we have is to take the sequence, and in this case, these are unphased genomes, so both haplotypes are more or less put together, if you will. And we we take sliding windows moving down the scaffolds every 100 kilobases, and we look at the number of SNPs, single nucleotide polymorphisms, which give us an idea of the diversity within that window. And you see two types of diversity here. You see the lower line in red, which is the natural within species variation. But when we have two different haplotypes, that is one part of the chromosome from one species, another from the other, now we can see much higher frequency of variation. And so we use this, and we can actually go down the chromosomes and determine at any point along those whether it's pure mandarin-mandarin or pure pomelo-pomelo, or one haplotype comes from mandarin and the other comes from pomelo. And in so doing this, we can, here's an example of what it looks like. So on the left-hand side is a panel where we're comparing pomelos with each other, and you'll see the natural variation as you would expect within the species, but it doesn't get above this threshold. On the right-hand side is a panel where we were looking at Ponkan mandarin and willow leaf mandarin, and you see a stretch up until about 18 megabases on scaffold number 6, and then suddenly it jumps up. This is telling us that in that region of that genome, there's two haplotypes. It's mandarin and pomelo together, not mandarin and mandarin. And so when we do this now, we can, in a comprehensive way, get an idea of the proportion of the different species that are contributing to these different kinds of citrus. So much to our surprise, we found that among these 8 genomes, none of them were pure mandarin. Our expectation that Ponkan or Suntara was pure mandarin was destroyed because a good part of it comes from pomelo, introgression of pomelo genome into that reticulata. So the first two you'll see are painted green and green, and that's telling us that both haplotypes are from maxima. Those are pure pomelos. You can see Ponkan mandarin third. Much of it is mandarin-mandarin, but there's a proportion at the top that's painted blue to indicate that it's a mixture of mandarin and pomelo, and a small sliver of green in there telling us that actually a part of Ponkan mandarin has both haplotypes coming from Citrus maxima, from the pomelo. Hardly a pure mandarin. The same is true for willow leaf. In the 5th position, you can see sweet orange, and much of it is painted blue, which tells us that most of the genome of sweet orange is a mix of mandarin and pomelo. There is a proportion that's in red, telling us that that part of the genome is both from mandarin, but also in sweet orange, there's a 6% more or less amount of that genome that's maxima, pomelo, pomelo. We look at the next one, which is clementine, and go beyond that. Now you'll see SSO, which is standard sour orange, entirely painted blue. That tells us that sour orange is a true F1 hybrid between some pure mandarin and some pure pomelo thousands and thousands of years ago. Next. We can take this and we can look on a chromosome by chromosome basis. Each of these 9 panels represents one of the basic chromosome numbers of citrus, and you can look at the different cultivars and basically understand the genomic architecture of these plants at any given chromosome. Next. So we're missing one photo, but that's okay. We could divide our 8 genomes into different clades based on their chloroplast, and we can see one which is based on reticulata, another based on the maxima. On the bottom right-hand side, you'll see a photo of a fruit. There was another on the left of the external part of that fruit. And our friends in China published a paper at the same time, and they were calling this thing Citrus reticulata. And one of the beauties of the modern world is when somebody publishes a paper in genomics, the data must be made public. So when we publish, or you publish, or the Chinese publish, we have access to an ocean of information. And we took the information that the Chinese published on this thing, and we looked at it. And here in panel C there's principal component analysis. On the lower left-hand side, we have the mandarin group. On the lower right-hand side are the pomelo groups. In between there are sweet orange and sour orange, and way up on the top you see a dot that's completely separated from all of these. That's this thing that the Chinese were trying to say was Citrus reticulata. It's not. It's a unique, pure species called Citrus manchungensis, and it's ancient. It's nearly as old as Poncirus trifoliata. Next, please. So we published this in 2014. Again, it was an international team with people from Spain, Italy, France, Brazil, and the United States. I think that's all. But an international group, and we published in Nature Biotechnology. Again, it was one of the first publications on citrus genomes. We learned, whoops, go back please, that our mandarins, all of the mandarins that we had looked at were derived from these two ancestral species, reticulata and maxima. And pomelo admixture was found in all of these, and that thing, funny-looking citrus, Manchung Yegang, thought to be a mandarin, actually was a unique and distinct species. Next, please. Time passed, technology improved. We were able to do more and more genome sequencing more rapidly, more inexpensively, as well as many other people in the world. And we had a larger collection of genomes now that we began to look at. And they ranged from some materials coming from India, China, Southeast Asia, and all the way down on the lower right-hand side, even from Australia, and extending up through Taiwan, Okinawa, and into Japan. So we looked at these genome sequences, and we saw nothing. Okay, here we are painting the same kind of compilation that we did earlier with just the mandarins. On the top we had 4 different citrons, all of which were pure citron and citron. Where you see the solid green bars, those were pure pomelo. But in between those you'll see LIM, which is lemon, true commercial lemon, and you'll see half of that genome is painted light blue, which is a mixture of citron and pomelo, Citrus medica and Citrus maxima. The other half is painted orange, which is a hybrid of citron and mandarin. This tells us that lemon is a tri-specific hybrid, and we know now that lemon originated by a cross of sour orange by citron. Below that below that you'll see LMA, that's Citrus limonia. You might think rough lemon, you might think Rangpur lime, and those you can see are painted solid orange. It's a pure F1 hybrid of mandarin by citron. And we can continue. When you go to the bottom, you'll see almost all solid red. That was the closest thing we had at that time to pure reticulata. And as you go up, you see less red and more blue, indicating greater and greater introgression of pomelo into this mandarin background. Some of those things that are up there include Ponkan mandarin, willow leaf mandarin, king, satsuma, sweet orange, and then you get to the sour orange. So you can see, again, if we want to look at this at a chromosomal base, let's backtrack a little bit. We looked at the mandarins and we divided them into 3 types. Type 1 are the pure Citrus reticulata, and what we found there was Tachibana from Japan, Sunchu Shakat from China, and several others from China. No introgression of pomelo. And then we moved through the others as we talked about before. You can see again the limes and the lemons, the pure pomelo, the pure citron, and so on. And if we want to, we can look at this. Okay, there's probably a slide missing. It doesn't matter. It's a confusing slide. Let's get to the point of the story. Above the top, you have the pure biological species of citrus. Kumquat, Citrus micrantha, citron, Citrus medica, Citrus reticulata, and Citrus maxima. You can see our sour orange originating as an F1 hybrid of mandarin by Pumelo. You can see further introgression of Pumelo leading to those type 2 mandarins, and further introgression to the later admixture, the modern hybrid mandarins that we know. Sweet orange comes from that. Grapefruit is a hybrid of sweet orange by Pumelo. You can see again the lemon as a pure hybrid of sour orange and citron, Rankper lime and rough lemon as hybrids of different mandarins with different citrons but the same specific contributions. Next. So let's talk briefly about evolution. And to talk about evolution, we need to keep the perspective of time in mind. We're talking about evolution across the last 8 million years of the Earth's history. And if we look at human evolution across nearly that same period of time, you can go back and see what we once were, perhaps. And we have to keep that in mind as we're talking about the evolution of citrus, that what we see today is not what existed over time. It's the results of evolution, diversification of these plants. Another important part of the story, as I mentioned, is plate tectonics, the Indian plate crashing into the Eurasian plate, causing an uplift of the land, resulting in the Himalayas, resulting in huge changes over several hundreds of thousands of years in the climate and the environment that led to rapid radiation, rapid speciation of many different plants and animals in the same area at the same time. So if we think about the center of origin of citrus, we can describe it as somewhat near that circle of citrus that you see right there. And you'll note it encompasses parts of northeastern India, northern Myanmar, and western Yunnan province in China. It so happens that a citrus fossil was found in a town in Yunnan province called Linchang. Linchang and Tinsukia, where we visited recently, are probably no more than 200 kilometers apart by air, maybe even less. I haven't actually gone on Google Earth and measured it, but it's very, very close. And my point is, this is the same neighborhood. It's the same neighborhood. When we traveled around northeastern India and I looked at the mountains and even sometimes looking at the people, I could imagine that I was in Yunnan, where I've visited many, many times. This is a photograph of a mountain in Yunnan province that's only 40 kilometers from this town where the fossil was found, 8-million-year-old citrus fossil, a place called Ilao-san. And in the foreground, you'll see modern citrus, grapefruit being produced there. But again, it's not that much different at all from northeastern India. It's all part of the same region, the same neighborhood. And very important things happen there for citrus. So there's our origin, and things moved in different directions, predominantly from west to east and southerly. And you can see some kinds of citrus found their way to Australia and evolved into what once was called microcitrus and eremocitrus. If you look at the upper right-hand panel, you'll see two different radiations, some of which is cut off from the slide. There was the Asian radiation that took place approximately 6 to 7 million years ago and gave rise to the species that we know as reticulata, maxima, medica, and so on. And a further radiation in Australia 4 million years ago, giving rise to those different species. What's interesting to us, there's one place where reticulata diversifies, and you can see Tachibana and Sunchu Sha Kat, two different mandarins. Remember earlier I said that Tachibana looked to be a pure mandarin, and at that time, based on the techniques we were using, it certainly appeared to be. But as we looked at the sequences, we saw something different, and we became very much more interested in citrus Tachibana. So this is another chapter of the story. Tachibana is culturally very important in Japan. There's another kind of citrus in Okinawa called shikwasha that's also very important to Japanese, particularly Okinawan culture. And shikwasha also has been used as a breeding parent for rootstocks. Many scholars speculated that these plants that appeared to be native in Japan actually were not native at all. They might have been introduced perhaps by humans, and they were adapted and they grew. They were feral populations. We didn't understand, sorry, go back. We didn't understand what was going on here, and we didn't understand fully their relationship to the mandarins from the mainland of Asia. So there were new genome sequences available from our group and also from Huazhong Agricultural University. And we began a simple study. One of my colleagues, Dan Roxar, is an adjunct professor at the Okinawa Institute of Science and Technology. And while he was there, the Okinawan people were saying, hey, we have lots of citrus growing wild here in the forest. And Dan said, well, let's sequence them. Let's just see what we find. And it turned into a very interesting story. Go to the next slide, please. And we published this story in 2021. We talked about the diversification of mandarin citrus by hybrid speciation and the importance of apomixis, the ability of citrus plants to reproduce true to type from seed through nucellar embryony. There's Tachibana next to a Japanese temple, and again, these two types, the Shikwasha and Tachibana. There was also a third group that was called Yukunibu, which were a number of interesting different kinds of hybrids, the origin of which no one understood at all. And so we sequenced 69 new genomes and began to study their relationships. And what we found was a previously undescribed species, something called Citrus ryukuensis. It's a very small fruit. You can see the fruit there peeled. You get an idea how small it is because that's my thumb.
Recording in progress.
That's my thumb in the photograph. Very small, but very important role this species played. There's a tree of ryukuensis on the upper right-hand side. You can see the seeds of it. They're monoembryonic compared to Shikwasha, which is polyembryonic. We also sequenced something that we called RK3, which we found to be extremely closely related to a mandarin that was sequenced on the mainland of China. So how did this mandarin come to Okinawa, and what was its role in this story I'm telling? Next, please. So again, we used principal component analysis looking at these genomes. In the first panel, you can see the mandarins on the lower right. Citrus ryukuensis separated, several individuals of the species separated. Sorry, mandarins on the left, ryukuensis on the right. In between were Tachibana and Shikwasha, telling us that these things were hybrids of mandarin onto ryukuensis, this previously undescribed species. You can see the oranges above that and the pomelos separate. In the next panel, what we're looking at there on the left-hand side, on the bottom, are what we call common mandarins. We analyzed sequences that our Chinese friends published and concluded that there was a separate subspecies of mandarin in China that was not recognized, but sufficiently distinct that we could have called it a new species. And we found some hybrids between those. Again, ryukuensis is on the far right-hand side, and you can see that Shikwasha lines up, indicating it's a hybrid of ryukuensis by the common mandarins. Tachibana, by contrast, is ryukuensis by hybrids between these two subgroups of reticulata. Next. So we have now a greater understanding of the diversity in mandarins. All the mandarins we know in the world are comprised of 4 different species, or 3 species, 1 and 2 subspecies: Pomelo, Manchung wild mandarin, the common mandarin, and the Ryukyu wild mandarin. The common mandarins are complex admixtures of those 2 subspecies of mandarin plus Pomelo. Tachibana are hybrids of those, and those funny-looking citrus that we saw actually are of much more recent origin. Other types were introduced into Okinawa by man, and a collection of new hybrids arose as a result of that. So, Citrus reucoensis is a new species. It has a distinctive nuclear and chloroplast genome. It has the lowest heterozygosity among all the mandarins, a sign that it is, in fact, a true species. It's monoembryonic. All of those pure species I showed much earlier, pomelo, pure mandarin, citron, all produce monoembryonic seeds, an important attribute. But others are polyembryonic, and the polyembryony has been a very important driver in the domestication of citrus fruit. Incidentally, these types diverged from the ancient Asian mandarins 2 to 3 million years ago. So the idea that Tachibana and Shakwasha were introduced by humans is not at all true. Next, please. This reiterates the origins. We can go on to the next. So there is this characteristic of nucellar embryony, the ability of citrus to produce multiple embryos in a seed that are identical to the maternal parent. And it's the result of a mite insertion in a promoter for this specific gene. This mutation is found in all of the commercially important citrus that we know. Oranges, grapefruit, sour orange, lemon. And why is it important? You can imagine that there was at one time a very first sweet orange tree, for example. And if that tree did not have the ability to reproduce itself true to type, there would have been one tree on the planet. And the chances of humans finding that one tree really close to zero. But because it could reproduce, there would be small groves identical, and people eventually found these things. And so this was a critical driver for domestication. So that mutation occurred about 1.2 million years ago in the Manchang yeju, that other subspecies of mandarin, and it's moved throughout all of citrus over time. Now you can see the red star there. That's where these mandarins originated in China, and it's difficult to see, but Okinawa is out in the middle of the water. So how did these guys get together to have a baby? Next slide, please. If you go back in time, 7 to 8 million years ago, the eastern edge of Asia is where you see it here on the upper left-hand panel. There was no water there. It was land all the way. So very easy for those species, those plants, to move and spread. 2 to 5 million years ago, the sea levels rose and it was isolated from the mainland. So diversification, evolution, things separated. In the time frame we're looking at in this tree here, 1.3 to 1.7 million years ago, the lands were connected again. So those plants were able to be together because they were growing in the same place. And here we are in the modern time. Next, please. So why is this important? Well, on the right-hand side, you can see me standing in the COVID days next to a sweet orange tree on a Shakwasha hybrid rootstock, extremely tolerant of HLB. So it's important to understand these things. It's important for genomics to be able to look at the phytonutrients and these kinds of things to develop improved, more nutritious citrus. Next. However, as I mentioned at the beginning, nearly all the genomic information we have is excluding information on the native species of India. It's a missing piece of the puzzle. There are unique and understudied accessions, like Citrus indica, for example. We know one clone of Citrus indica outside of India. It's in the collection in California. It's in the collection in Spain, in Italy, in Corsica, probably in China. It's all the same clone. What is the diversity that we can find in indica? We were privileged to be able to visit some indica trees in the forest in northeast India just a few days ago. It's a missing part of the story. Citrus latipes, another native of India, 1 or 2 clones distributed throughout the world, but genetic diversity here. And we don't have genome sequences of these things. We don't fully understand their relationships to all the other kinds of citrus that we've studied. I'm sure it's a very interesting and important story. We have populations of other well-studied species we can find here in India, the citrons, many citrons. Dr. Chandrika published several years ago on 3 subpopulations of citron. They excluded anything from India because the genome sequences just weren't available. What we find in India, are they the same or are they different? Is it greater? Do we have greater diversity than we know of? Citrus cavalieri, also known as Citrus ichengensis. We heard it grows wild in Nagaland. We know it grows wild in Yunnan. We know it grows wild in central China. What are the relationships of these populations to each other? Important questions that we don't know. So the next chapter of this international collaboration must, not should, must include the native Indian germplasm, the sequences from native Indian germplasm. We hope to be able to work together and write the next chapter of the story together with Indian scientists. We've had a long history of doing that. Our work in Japan was published together with Japanese scientists. It's important that we collaborate and finish writing this book on the origin and evolution. But why is this important? Aside from being an academic exercise, in and of itself, that's important. But I work as a citrus breeder, and I work in the state of Florida where citrus greening disease has eliminated 85% of our production in 15 years. I'm working to make a genetic solution to this. We have a population that we made, and it's small. You probably can't see it very clearly here. But we made a cross between one clone of Latipus that we have with Clementine mandarin. And we planted out over 500 trees. The population is large, so we could do mapping. We could do QTL mapping. We could do genome-wide association studies. What we found when we analyzed these trees, some of the trees after 5 and 6 years in the field, not some, the majority of the trees, we still cannot find Liberibacter in those trees. Is this Latipus a source of resistance? What is the nature of that resistance? It's a critical question. These hybrids are fruiting. We have second generation hybrids made to see that we cannot perhaps recreate the sweet orange, recreate our commercially important varieties using genome information to reconstruct these genomes with resistance to HLB. So it's a very important goal. And with that, I'll thank you for your attention. A photo of our first released variety called Sugar Bell, also very tolerant of citrus Huanglongbing disease. So, thank you.
Thank you so much, sir, for giving insights in untangling the citrus taxonomy, and we hope things will work out. And now I request Dr. David Karp to give his lecture on modern citrus cultivars descriptive database.
Not the boarding pass. This one right here. Yes. Open. All right. So this doesn't work to advance it? No. Well, it won't work for me then, probably too. Yep. Okay.
Recording in progress.
Okay. It works? Fantastic. Okay. Let me just make sure that. Where do I have to point it? Okay, that makes sense. Okay. Well, I'd like to start by thanking our hosts from ICAR. We've been here for 18 days. This is the trip of a lifetime. I've been dreaming of this for at least the last 20 years. I've got notes from the late 1990s where I was seeking to form the basis for a trip such as this. All the pieces came together this year, and boy, are we excited. We learned so much. We met so many interesting people. We formed plans for the future, so I am delighted to be here. It's tough following Fred Gmitter because he's the Mick Jagger of citrus scientists, but I'll do my best. I'm here to talk to you today about the Modern Citrus Cultivars Descriptive Database, which is part of the infrastructure of pomology of fruit science. In order to understand what it is I'm talking about, I'm going to back up a little bit so that you can understand where it started and what its context is. Basically, pomology, descriptive pomology involves descriptions of fruits and the trees, including information such as who bred it, where it originated, what's the pedigree, and now these days, what are the intellectual property rights, what's the nomenclature. I started out getting involved in this some 17 years ago in 2006 when I was recruited to serve as one of the contributors to the Register of New Fruit and Nut Cultivars. About the same time, I started working at the Citrus Variety Collection, building their website, describing some 1,100 accessions, and their love child was the Modern Citrus Cultivars Descriptive Database, which involves mostly intellectual property protected varieties, some of which are not readily available to anybody or to India or to us in California, but are nevertheless of very great interest because you want to know what people are doing around the world. If you're a breeder, if you're a grower, if you're a marketer. So I'll start with an overview of what the register is. From the name, you might think it's a government intellectual property register. No, it's not. It's not an official register of that sort. Its antecedents actually stretch back to the late 18th and early 19th century with the beginnings of descriptive pomology. It's a scientific publication. The register itself started based on various antecedents in 1944, long before UPOV, the Intellectual Property Global Union, or the International Code of Nomenclature for Cultivated Plants. It's published biennially every 2 years in Hort Science by the American Society for Horticultural Science. And the content is written by some 55 pomologists. It involves not just citrus, that's only a small portion of it, but as many as 100 different forms of fruit over the years. I'm currently the senior editor and with my co-editor Ksenia Gajic of Clemson University. It includes, for the most part, just fruits and nuts of North American origin that are present in North America, or, and this is a slippery slope, that are conceivably important for North Americans. It includes both public and private varieties, only cultivars introduced since 1920 and later, and a host of information including nomenclature, pedigrees, breeders, IP details, descriptions of trees and plants, and the fruits and nuts. So who's it for? Who's interested in this? Breeders, pomologists, farm advisors, nurseries, growers, intellectual property owners and managers, anybody interested in fruits and nuts will want to know this information. What cultivars have been introduced around the world? What are they all about? What's their reason for being? Along with detailed descriptions insofar as those are available. What's it for? You want to consult cultivar descriptions about scions and rootstocks, see what's new, find intellectual property details. It includes cultivars protected by various different intellectual property modes, find the pedigrees, see if a name has been used. If you're introducing something, according to the regulations, you can't be reusing a name. And find, this is the trickiest, the craziest part, find which trade names correspond to cultivar names. In much of the Western world these days, there's not just one name, but two. more than 80% of new cultivars that are introduced have an alphanumeric code, and this has changed radically just in the last 10 years. Very few people, even professionals, unless they're intellectual property attorneys, really understand what's going on there. What's not included in the register are exclusively ornamental cultivars, forage plants, wild germplasm, or unreleased breeding populations. Next slide, please. There are great challenges in the last 2 decades include a huge profusion of new fruit and nut cultivars, the globalization of breeding and trade, the privatization of breeding. So you can't necessarily plant something because it's not made available to you because somebody else owns the rights and won't give it to you. The challenge of whether you want to be descriptive or prescriptive, that is, tell what's going on or say what should be the case. There are new breeding technologies. There's resources of the internet, and which some people will say, oh, why do you even need to bother have one source brought together? Yes, you can sometimes find this yourself on the internet, but in many cases, I'd spend as much as a whole day just describing one cultivar, finding information that not that easy to dig up. Nobody teaches you this in school. So all of this information, remember, this is published in an article of about 60 pages in Hort Science every 2 years. Everything since 1944 is brought together in a searchable database that you can be in the middle of a mango orchard here in central India, and you can look up American mango. Remember, it's just mostly, for the most part, just North American varieties. But if you want to see what's the pedigree of Kino Mandarin, for example, you can look it up. Anybody with access, freely available to anybody with access to an internet connection. In 2016, we launched a program to bring everything online, and it wasn't easy because, unbelievably, the files had been lost. I ended up paying somebody $39,000 to go over an 800-page book and integrate it with all the files since 1944. But now it's online. And I'm going to give you, at the very end, I'm going to give you a handout with where you can find all the information I'm talking about. I've had to put together numerous, about 24 different appendices as to back up, just in order to do the job of compiling this information, just to take a look at one of them. For example, there's no resource for how to find and use intellectual property documents for plants. Why is that important? Not just for the intellectual property, but because they often contain descriptions of plant cultivars. Where can you find that information? You'd think that there would be a website somewhere that would tell you that. Well, there wasn't until I put this together. And it's easy to find, to search by country. India's not in there yet, even though I do have a good idea of what your IP website looks like. It was last presented to CCRI, which is an UPOV-based organization, just for signatories to the 77 countries signatory to the UPOV Convention. So, but I am going to add India and other countries with sui generis systems of intellectual property protection. So, finally, before getting to the modern citrus database, this was not my first rodeo putting together, updating the register, dragging it, kicking and screaming into the modern world. Starting about the same time in 2006, I started working with the UC Riverside Givaudan Citrus Variety Collection, helping to build their website, took over 100,000 photos. We put up 1,100, we being the directors, Tracy Kahn, Tony Siebert, Karen Trinell, and various others, put up 1,100 pages, gathering together information about all the citrus varieties that are in the collection. And that's readily available at citrusvariety.ucr.edu. And again, I'm going to provide a link at the end. So the love child of the register and the modern citrus and the UCR Citrus Variety Collection website was the Modern Citrus Cultivar Descriptive Database. I actually ended up putting together, just by chance, because I tried to download a list of all the citrus that's protected by UPOV in the Pluto database, and I realized that it didn't necessarily tell you. It would give you the name of a cultivar, but it wouldn't tell you, was it an orange or a lemon or a lime? What was it, a grapefruit? I thought, well, I'll just quickly annotate this. I thought it would take 3 hours. Guess what? It took the whole year, 2020. but I did put it together. It's useful because the citrus variety collection only has publicly available information. There's no way we could plant everything that's been developed around the world, because we'd have to come up with another $100 million to buy land and hire people, and we wouldn't be able to get the rights anyway, because a lot of these varieties are protected or not readily available, or would cost a lot to bring them in. So I started this in an attempt to just figure out what citrus has been developed around the world. I started annotating a spreadsheet from the Pluto database, and after 1 year, I'd pretty much finished with 1,200 varieties or so. Again, the citrus variety collection covers mostly publicly available cultivars. So in some ways, we're like a library that hasn't acquired any new books in 20-something years. Modern Citrus Cultivars Descriptive Database covers cultivars that in many cases can't be studied directly, but about which much information is available from sources I'll describe in a minute. Oops, sorry, I should have. This is what the website. It's on the University of California Riverside Citrus Variety Collection website, but it's its own section that describes. It's not in these cultivars are not necessarily in the citrus variety collection. This is everything else that's available around the world. And I'm not trying to reinvent the wheel, so I'm not including everything that's, for example, was in the Citrus Industry Volume 1, the Citrus Bible from 1967, or James Sawn's Citrus Varieties of the World from 2000 or 2001. But it's still got a lot of stuff in there. Ooh, that's a website not familiar with me. That's what it looks like, except it would stretch like from here to the other end of the room. So it's a huge Excel file. It's not beach reading, but it's eminently searchable. It's a powerful tool if you want to see everything ever bred by Fred Gmitter, for example, or Mike Roos of the University of California, Riverside, or see all the kumquats that have been developed in the past 40 years. It's eminently searchable by scion or rootstock or ornamental. It does include ornamentals. So you can search by 25 different categories. And the most important thing is the description. Let's see. Oh, it includes not anything in the genera Citrus, Pancyrus, Fortunella, Microcitrus, and Remocitrus, and their hybrids. So what's in it? Well, nomenclature, and there are 7 different categories there. Who knew that there would be? I was just counting them up the other day and realized, oh my God, our world is a lot more complex. At least this provides a reference source if you want to look something up. You can see, for example, the name of a Chinese or Israeli cultivar in their native script. And believe me, that's critically important. Trademarks and which trademarks correspond to which cultivar. Common name and according to a systematic method, botanical names in 5 categories, intellectual property details, the breeders, their organizations, which country they originated, descriptions of both scions, rootstocks, and ornamentals, and then so the descriptions will just look like, oops. Yeah. Descriptions are like 50 to 250 words. So sometimes there's not very much information available and there's a blank line there. But I've really beaten the bushes and tried to come up with everything that I could over the course of the year and updated it regularly every week since then. This was the most difficult part, compiling the descriptions from numerous sources that you'll see which are those sources in just a minute. And very importantly, and I didn't I went back at the very end and said, Jesus, I'm going back to look for where I got this information because I've forgotten it. So I went back and put in up to 10 different sources, or always with hyperlinks when available. So you don't have to trust what I did. Maybe I didn't translate a Chinese document properly. You can go back and see what it is with the citation and hyperlinks. So where did all this information come from? Well, from plant patent and plant variety protection documents from various countries around the world provide descriptions along with their plant breeders' rights documents, scientific literature, release notes, the register, International Society of Citriculture proceedings, translations of foreign material, descriptions from citrus books. I've got over 300 at this point in my library, notes from 30 years of citrus research, brochures from commercial countries, information sent by breeders in many cases. And there is an opportunity to include recent Indian citrus cultivars. I'd like to talk to you and your director, find out who would be the best person to collaborate with me if somebody is willing to do so, to include Indian cultivars as they're developed. So I mentioned that I've presented a huge amount of information. What I'd like to do is hand out, so there is an easy way of getting to all 7 of these resources, and that is, the easiest way you can remember, even if you don't get a copy of this, Google or search for in your browser American Pomological Society, and then fruit and nut cultivars. It's right up here. And it's easy. All 7 of these will appear. They'll be these links will be constantly updated, hopefully. And easy peasy. I also have a copy of a description, just one, because it hasn't appeared yet. It's in press for the next Acta Horticulturae, based on the International Citrus Congress held in Mersin, Turkey last November. An article describing the modern citrus cultivars descriptive database. I'd be happy to give that to your director or to any of you that would like to consult it. So with that, let's move on to the next presentation. Thanks for your attention.
Thank you so much, sir. And we have another great presentation who is trying to make sense out of complex world of citrus. So now I request Dr. Chandrika Ramadugu to deliver a talk on citrus honglongbing. Can Indian germplasm provide solutions?
Going to move a little this side so you can see me. Thank you for the opportunity to speak with all of you. I was here at your international conference in October and enjoyed it a lot. Many people ask me, didn't you go back to USA? Are you still here? No, I did go back, and I do have a job there and came back here recently because I was very fortunate to get included in this group of 6 people that went to Northeast India to see the citrus. And my primary interest is citrus honglongbing, and that is the reason why I wanted to go to Northeast India. I'll explain to you why. So Georgios, I'll finish in 10 minutes, so you have 10 minutes, okay? You don't have to rush. No, that's okay. Do I forward? Yeah, please go ahead. Yeah, my name is Chandrika Ramadugu. I'm a product of Indian Institute of Science, Bangalore, and right now I work on citrus genetics, citrus breeding, and disease resistance. Next slide, please. Oh, here, okay. Can you somehow bring it down so my titles are not cut? No? Okay, that's fine. So citriculture, or cultivating citrus in many countries, is impacted because of honglongbing problem. Okay? Honglongbing, or citrus greening, is caused by a bacterium that cannot be cultured and spread by the Asian citrus psyllid. As Fred Gemeter mentioned earlier, in some parts of the US, that is in Florida, a lot of the citrus, almost 80% of the citrus, is affected because of the honglongbing problem.
Recording in progress.
Oh, I'm sorry. Did I do that? No. Okay. So this disease, HLB, was first reported in India 100 years ago, 1912. Okay, that's when they called it decline. And we saw that even now many people are calling it decline, and many people think that it's separate from HLB, but we think that it is part of HLB. Maybe it has something else too, but definitely HLB. And we saw this decline everywhere in India, except in some hills of Assam, where about 4,500 feet elevation, and there was no HLB, although the pathogen was there in other areas. So we don't really understand why. And Northeast India, with its rich genetic diversity, we think it may provide very valuable resources to develop resistance to this disease issue. So in the Western Hemisphere, HLB is present in the United States, in Brazil, in Mexico, in all countries in between, okay? So it is a big problem. The thing is, there is no cure for this disease, so development of long-term solutions is needed. I just want to give you an update and show you this figure. This is published recently in some farmers journal, growers journal, citrus industry. So the citrus bearing acreage in Florida has been declining from 2003 to 2022. You can see how it's going down. Although there are other reasons, HLB is the main reason. Although a lot of research has gone into this, there are very few things that have provided solutions so far. There may be several that are in the offing, and we may have fantastic solutions soon, but we don't have them in our hand yet. Okay? One of the things that is working is growing citrus under protective structure. So here you can see field trees that are covered by a big mesh enclosure. I don't know if you can see that. And the size of the mesh thing is much smaller than the psyllid. Okay, the psyllid is small, this is smaller, so the insect cannot go inside, and hence there is no disease. Very simple. So it works, but then it is quite expensive to build these structures, and you can't possibly do it for the whole country or the whole state. It works for research organizations only. So another method that they are trying now is the antibiotic treatment to manage the disease. So they have been injecting oxytetracycline to the trunks of the tree. So they make a hole in the tree and inject the antibiotic, and they do it second year also. And temporarily it works well, I think, but I don't know how many holes the plant can take and what will be the long-term effect of having that antibiotic on human health. So this is also probably a temporary solution. Some of us geneticists and breeders like Fred and myself, we believe that if we do the right type of breeding experiment, maybe we can have genetically resistant plants, and that will be a ecologically sustainable method of cultivation. So I'm from UC Riverside, California. It shows where we are. That's California right there, and you can see that. Anyway, the red stars show where we are. And David Karp, who just talked now, he talked about the variety collection in Riverside. I had some pamphlets, I forgot to bring them. I apologize. Tomorrow I'll bring some and give it to you. So we have a collection with about 1,200 plants or so, 1,200 varieties. Many are from India and from all over the world. This collection is about 100 years old. What I want to tell you is that those are available to people all over. You just have to get the right permits and ask the right people. There is some procedure, so we should all make use of that. So we have, again, we are afraid that we will lose everything to HLB, so we have copies of these plants in quarantine, and that emphasizes the need to protect whatever you have, whether it is in India or US or Brazil. We're also building extra cup structures so that we have backup of these citrus accessions, because we know the disease is going to come there. We try our level best to control the insects, but it's only a matter of time. And Georgios is going to talk to you next. He is going to tell you about the pathogen tested budwood that will be provided from these, and that is a very important aspect of citriculture. So this is a picture showing different varieties in our collection. It's pretty impressive. The reason I showed you all this is because we conducted a field experiment several years ago, trying to see if there are some citrus varieties that have resistance to HLB, and there were not many. Okay, but then we found that some citrus germplasm from Australia had resistance to citrus HLB. So these are the ones we used in our breeding program. Now this is called Eremocitrus or desert lime, and the other one that looks like a finger is called finger lime or microcitrus. These have either total resistance or very high level of tolerance. And the advantage is that they are sexually compatible with citrus, so you can make hybrids. And if the hybrids are resistant, then you can do back crosses and generate something that has both resistance and some bears a fruit that is edible. Okay. Now, why do breeding? Because no one likes to do breeding of trees. You know, it takes forever, right? As you all know, in citrus there is genetic vulnerability. Like if you have grapefruit, all grapefruits are derived from one variety, so they'll be genetically similar. Although the flesh color is different, the rind color is different, genetically they're very similar. So some situation like that is not good, especially when new diseases come or when any bad disease comes. So you need to do genetic enhancement or broadening the genetic base, which can be done using other germplasm that's not typically used. Okay? Then we have to do the evaluation. This shows some of my hybrids. The ones in the top panel are the susceptible hybrids. The ones in the bottom panel are resistant hybrids. In California, we have lots of restrictions, so we can only do the experiments in a protected greenhouse. So we have many limitations. So I want to have free access where I can put 100 plants and do evaluation of Indian germplasm right here in India. So I want to have collaborations. This is just to show you that the hybrids generated using this weird germplasm is not all bad. I do have bad ones, but you can get good ones, and this is in second generation. So there are many that have edible fruits. They do have a little bit of off flavor that you can taste. Hopefully it will go, and hopefully if you have enough hybrids, you can screen for the ones that are best. And we are still screening these for HLB because that is a long process and that is very important to do. This one shows a pomelo. As you know, pomelos are grown as seedling plants, and this pomelo was found in Coorg, where we got HLB and everything was destroyed. All the Coorg mandarin was gone. But there were some pomelo trees that were 50 to 60 years old that survived, that didn't die. So this pomelo was brought to the US, and my colleagues have sequenced it, and we are trying to see if we can use that. So this is the same pomelo tree. Basically, we think there are sources of HLB tolerance or resistance in native Indian germplasm. We are primarily interested in a couple of accessions. One is Kasi papida, not just one accession. You probably have multiple accessions. Like Fred mentioned, Kasi papida was highly tolerant. So crossing Kasi papida with some of our hybrids may also be useful. And another one is Citrus medica. The thing is, we don't know if it's really resistant to the disease, or it is just that the place where Citrus medica is there, there is no psyllid, and hence there is no disease kind of thing. Because although we are able to get resistant hybrids using the Australian citrus, it is essential to develop citrus varieties that suit different environments, different types of fruit. Like, they want Kasi mandarin type. Some people want Kino kind of mandarin. You want Nagpur kind of mandarin, right? So in the US, in Brazil, in Argentina, everywhere they want different types of fruits. So we want to use as many sources of resistance as possible and see what we can come up with. So that's it. Thank you.
Thank you, madam, for your nice presentation. I hope the next presentation will provide some solutions or way forward to control this HLB. So now I request Dr. Georgios Vidalakis to give presentation on Citrus Clonal Protection Program, a model system for citrus germplasm and nursery stock disease protection.
Well, it's my turn to thank you all for receiving us to your beautiful country and the beautiful institute. And it has been truly an amazing visit and experience to see and visit all the scientists and farmers and
Recording in progress.
How you grow citrus in India and how important is your country on, as you heard already, in evolution on citrus. So my role in this team is not for the preservation of the germplasm, but to facilitate the movement of the germplasm. As you know, when we move citrus materials, we may move diseases and pathogens along them. So my role at the University of California, Riverside, is to make sure that materials that they are coming into the state are tested for diseases and they're also undergone therapy, and then they can be redistributed so to the industry or the scientists. So if in the future we come up with a collaborative project with India, materials will be handled by the university in collaboration with the federal program. We have two citrus germplasm programs in California, the University of California that we're focusing primarily on commercial citrus, and the USDA National Clonal Germplasm Repository for Citrus and Dates. And Dr. Robert Kruger, which some of you have met and interacted, is responsible for that program. So between the two of us, the University State program and the National USDA program, we will make sure that whatever project you bright minds come to execute, we will make sure it will be executed safely in terms of disease and pathogens. So as Fred said, the story I'm going to tell you today is not my story, but a story of many, many people from around the world. We have great collaborations throughout the United States and many different countries. So the words cut off in the slide says, what do we do? I give you a very brief introduction about California citrus. So this is the state of California. You saw a map already. We are growing citrus in 5 climatic zones. Desert in the south, limes, grapefruits, coastal areas, lemons, inland empire, the light blue color, where Riverside is, navel oranges, mandarins, and so on. But the heart of the industry is the pink color in the center, in the Central Valley, where they grow everything, and it's great, the greatest acreage. One last area of production of citrus is up north in the Sacramento area, primarily early maturing satsumas to avoid the freezing temperatures. You see a breakdown of the acreage. We're still a navel orange industry like we were in the early 1800s, with mandarins growing in significance in acreage. Very important to remember that California is a fresh fruit production industry in comparison to Florida or Brazil, that are primarily processed citrus fruit industries. We produce fruit for the first market. Florida is producing fruit for juice. California citrus is valuable. When I started with the industry as a student in the year 2000, the industry was less than $1 billion. It was $0.7 billion in value. You see today it's close to $4 billion. It has quadrupled in size, has exploded. The basis of the industry, as all of you know, is a healthy nursery production. We're producing roughly, we're estimating, and that's why I put an animation there. As Dr. Ghosh mentioned earlier, he visited our program this summer with the Asian Development Bank, and there is plans to make a serious investment on production of clean germplasm for India. So if I have, and I will try to give you a few tips throughout my presentation from our experience. Make sure you have a mechanism to record the activities and know exactly how many trees you're propagating. Unfortunately, I don't know, for unknown reason, nobody could give me an answer. In California, we don't know how many trees our nurseries are propagating. We're working now with the state to be able to record. Some of it has to do maybe with business secrets and all these things, but come up with an idea how many trees our nurseries actually propagating, so you can have, as you will see later in my presentation, you need to be able to measure it so you can improve it if there is problems, any program. So what do we do? What are the challenges we're facing? Craft transmissible diseases of citrus, including, of course, HLB. We don't have time to go in detail in citrus diseases, but just an idea there on the right-hand side. A viroid disease important in California because we're using primarily trifoliate and trifoliate rootstocks. It's Exocortis. A virus disease. Still today there is quick decline in California. Some old orchards are still on sour orange. And of course HLB that so far has been detected in urban areas, only in backyard trees, but it's a matter of time when we're going to find it in our orchards because the vector has been spreading very effectively in the state of California. And of course, in the case of citrus, it's not like corn or tomatoes or that you lose this year's yield, but it's a very complex loss, right? You're losing the original investment to buy the trees and plant the trees, and then you have to dig them up and replace them, and it's a multi-layered cost that it's a very good idea to avoid it, and meaning you start clean and then you can maintain your production. So what do we do to protect California from graft transmissible diseases? We have rules. The title is cut on the top there. We have federal rules. Because citrus did not originate in the United States, like in your case, we don't have wild citrus, came in the 1950s, the federal government passed a law that says citrus plant, citrus green material, not fruit, cannot enter the country. We're talking about propagative materials. Unless if specific individuals, like in the case of Dr. Robert Kruger with USDA and Dr. Taylor Smith in the state of Florida, and myself in the University of California, we are the three people that were permitted by the federal government to import material into the United States. So it's very important for you also, Dr. Ghosh, when you set up your system, make sure you bottleneck it to the experts, and then from there you can control the movement and so on. We also have state rules that they usually follow the federal rules. The state of California has its own internal quarantines and so on. And we also have a clean stock program for the nurseries. And if we have time, I'm going to go over it really quickly at the end of the presentation. We operate at the exclusion level, programs like ours. You prevent a disease to enter in your system, then you don't have to worry about it. Our program roughly costs $1.4 million a year. When a disease enters an area, you attack it as fast as you can to eradicate it. We spend over $45 million a year to try to chase the Huanglongbing, one disease. You're excluding all diseases with $1.4 million a year. You're spending $45 million to change one disease. And then when you have to live with a disease, like in the case of HLB in Florida, then the cost of those programs are in the billions of dollars. So it's an absolutely wise investment in terms of prevention programs of pathogen-tested propagative materials for citrus. I think someone activated a function to write on the screen and the remote doesn't work anymore. If you can click somewhere. Okay. Good. So how does it work? We don't control the environment, but we are controlling the content of pathogens, so we make sure we eliminate pathogens from the germplasm, and we make available the citrus varieties to the industry and the scientists. That's how we disrupt the disease triangle. And here is a very quick historical overview that I came up just for the presentation. In the early days, we were dealing with the cirrhosis disease because most of the plants were from infected material, and the solution was Nuceller seedlings, true to type seed, because cirrhosis is not seed transmitted. The industry used Nuceller seedlings to clean the industry. A lot of this going on now in Northeast India with cassie mandarin. People can get clean trees from cassie mandarin, from seeds of cassie mandarin. If you can click for the next slide, or the next. But Nuceller seedlings have issues with Phytophthora. We saw some of those problems in our visit, and there are fruit quality issues. Then we switched to sour orange rootstocks. But then sour orange rootstocks have tristasia decline. And then we switched to trifoliate and trifoliate hybrids. But these are sensitive to viroids, as we discussed earlier. And who knows what rootstock our breeders will come up with to deal with HLB. So if in any of those turns you don't have a source of clean stock, you cannot make the transition safely. It's almost like exchanging one problem with another. If you switch to trifoliate orange rootstocks, and then you start propagating scions that they have exocortis viroid, you're going to start dying and dying and declining trees. So it's very, very important to have these programs so they can provide the industry. Next slide, please. With clean stock, so you can have flexibility to adapt in new conditions. Our program is very old. It goes back to the 1930s. Next slide, please. And the original discovery that the cirrhosis disease is graft transmissible. We have a long, this slide is fine. We have a long history. Every 20 years or so, the program evolves. The most important evolution of our program happened in 2009 when the federal government realized that our programs are important and united clean plant programs for many different commodities throughout the country under the National Clean Plant Network. And the legislator, the lawmakers realized that they don't have to reinvent the wheel, but to connect existing programs that they were performing research and services in diagnostics and therapy and distribution of clean material. So we didn't make new investments on new centers. We took existing centers, like your institute is positioned excellent in this, and we came together in a network to serve the industry. And we have citrus network and sweet potato network and roses and so on, all the vegetative propagated cultivars, or I should say crops. And that's a footprint of the different networks. Citrus is the orange dots, primarily in the south part of the country. So that's another model maybe you can create. You have a few centralized experts like your institute, and then you have regional centers that they can maybe not do the whole thing that you're doing, SUTIP grafting and indexing. They can just have maintenance and retesting, because different states, in our case, have different needs, different size of industries. Centralization is a good thing, but it needs to be also spread out so they can serve the local needs. Our program is highly collaborative. In order to be accepted by the regulators and by the scientists and by the industry, they have to be invested in our program, or a program in India that you're going to establish. It cannot be only one institute say this is the way to go and this is what we need to do. They have to have, as we say in US, buy-in and invested interest. Can you click the next slide? Okay. So our program does two things. We make sure we bring material in, we test it for diseases, and we do therapy. and we also maintain the mother trees and we distribute materials. In a single slide, next slide please. Oh. The program looks this way. On the left side of the red line is the quarantine in Southern California and Riverside. On the right side is the foundation facility in the Central Valley. After the material has been therapied and tested negative for all known graft transmissible diseases, it moves in the heart of the industry. You remember the industry primarily is in the Central Valley, and from there find its way to all its different usages. In terms of quarantine, we have the molecular laboratory in the campus at UC Riverside, and we have our quarantine greenhouses off campus, isolated in a corner of a city, surrounded by hills. And on the back of the greenhouses, the little home there, that's the original building of the 1907 citrus experiment station when you visited this summer, Dr. Goss. Sutip grafting, I saw in your earlier presentation you are performing it, so no reason to spend too much time. When we receive material, we'll cultivate it in vitro. Within 2 weeks, the bud would flash. That's the picture on the right. To save some time, we are no reason to go through the video. This video is available online if you Google citrus microscope and fruit mentor. It's a gentleman that helped us to produce this video, citrus microscope and fruit mentor. You can see very nicely how we perform sutip grafting. And after, which is essentially propagating a tree in a microscopic level inside the tube. After the tree, the scion on the rootstock has flashed, and that's what you see here, pulled out from the glass tube with forceps. We remove the roots and we propagate that little plant on the top of a mature rootstock grown in the greenhouse. The rootstock has been bent to break the apical dominance. And then we place the material in a growth chamber, and you can see how the young flashes early. At the early stages, the plants are covered with a plastic bag to stay humid, and then slowly the bag is removed, and you see how the young flash is pushing out. So if you go closely and look at the bud union area, you can see 3 colors. You see the old rootstock from the greenhouse on the lower part, then you see a little triangle of a lighter color, which is the rootstock from the glass tube, and then the upper part is the variety that was introduced from Japan or China or maybe in the future from India. When those tiny, tiny trees within a year more or less reach roughly this size, we have enough material to proceed to testing. The same way, if you go through a treatment for knock on wood and serious disease, after you go through chemo or surgery, the doctor doesn't send you home. You go back for checkups, right, to see was the treatment successful. So we do the same thing. We test for all known diseases, both in the greenhouse with bioindexing, which are indicators on the top table, and in the laboratory, which are the lab tests that all of you are familiar with PCR and gel electrophoresis and so on. The beauty of our programs and the importance for our programs, like it's how it's going to happen or it's happening with your institute, is that they are tied to research, right? The technologies that I started working with 23 years ago were developed in the 50s and 60s and 70s. But as you can see, the technologies keep evolving, right? So our generation, the past generations brought in the technologies of immunodiagnostics and PCRs. Our generation is bringing in the technologies of high throughput sequencing and so on. So it's, if programs like ours are not tied to research, and this is not my statement because of our program, it's been observed around the world, if they are not related to research institutions, they decline. And that has happened in many countries and states in the United States. Another very important element for their sustainability is financial support. In many, many countries and many, many states started programs in the 50s where it was the boom for these programs, but then for whatever reasons budgets were cut, and then the programs were starting and stopping and starting and stopping, and that was a death sentence. So another tip, Dr. Goss, is make sure you fight hard to have sustainable funding so you don't have to start and stop throughout the years. And by doing this process, As I told you, we're protecting against many, many diseases. There is a very long list of diseases that they are not present in California, and we don't want them to be present. Another interesting video online from Fruit Mentor is the 14 citrus diseases that we must avoid. And recently, we were able to digitalize completely our program, so I can see in the computer all the different steps and functions. So I ran an analysis to see, is there a specific variety that is more infected than others, or a specific country that maybe is has more diseases than others? And essentially, there is no pattern. It doesn't matter where the budwood is coming from. It doesn't matter what accession it is. Moving germplasm has high risk that it's going to move a pathogen. That's the reality of the, and especially if it doesn't come from another program, 99% of the times has different pathogens. So make sure that if germplasm is moving, is going through a program, because again, there is no pattern, there is no prediction other than if you move it and it was in the field, chances are that it has a pathogen. And this is very important because we, the scientists in our programs, we know what we're doing, but other people do not, and people that are not in the citrus world. Regardless of the laws and all our efforts and all the research and all the programs in California, a few months ago we discovered a virus in the Central Valley in a backyard home, the citrus vein clearing virus. And that was because the homeowner and so on, they didn't know what was going on, and we don't know exactly how it came there, but clearly this virus entered California. And the beauty of having this long history of a clean program in California, when the regulators and the industry called us, they asked us, are you testing for this virus? I said, yes, of course we do. Since it was characterized in 2012, we are testing with PCR. So we knew that our pipeline is clean. We knew that we did not introduce the virus and the nurseries do not have the virus. Very, very important for nursery production and industry image to the public to have programs like ours to address situations like that. Okay, I mentioned earlier to you that our industry has been growing and our program has been growing along with it. You see all our trends in terms of how many varieties and how much therapy we can perform, how it has an upwards motion. Also very, very important to build it in your program, room to grow, to help and support your industry or your research needs. Because if you build a program and you don't provide the service at the level that is required, then the trust of the users of the program declines, and then the program declines along with it. And this was my early comment: make sure you have a way to record your activities and evaluate your activities. So because now, as I told you, I can monitor the program just sitting in that chair, I can see that we were losing a lot of time. The bottlenecks in our production was the gray zone, which is growing plants, and the orange zone, which is performing diagnostics. Our target is to finish all our process within 2 years, 104 weeks. And you see in many cases we are passing the 104 weeks just by waiting for the plants to grow. So by monitoring our activities, we know where to spend time on research, and that's what we have been doing, how to improve our activities. And as a result, the different dots that you see in the graph is how the program was performing, let's say in the 80s and the 90s. And you see how the dots are spread all the way to 100 months or 200 months, and how we investing on research, going back, programs like this, because some countries or some people have ideas to privatize these programs and make them private, and the industry can do things better. Sure, the industry can do things very, very well, but programs like ours, they have to be accompanied by research to achieve this, to compress and improve our services so we can be very valuable to the industry. When all this part of the presentation was about our quarantine part of the program, then when the variety has been released from quarantine, tested negative for all diseases, moves to the foundation block. I have just a few slides for our foundation facilities. Protected foundation block. In California, we had the luxury, because we had all these programs since the 30s, we had the luxury to produce and cut budwood from outside grown trees from 1961 to 2007. An amazing run. an amazing resource to have in our hands for many decades, but since 2007 we had to transition only in screenhouse budwood. Close to 400 something varieties available in our foundation block. Very, very important, as also earlier from your video, Dr. Goss, that you do exactly the same thing with your institute. On the lower part is our chancellor, and the lady on the red shirt is the Secretary of Agriculture for State, Karen Ross. So very important to educate regulators, scientists, growers on the top about the value of our program. Full digitalization of the budwood distribution, because many times someone wrote the wrong number and then you have a lot of problems. Nobody writes anything anymore, everything is digital. We scan barcodes, we print labels, and also we can monitor our activities for improvement of the program. And because of all those activities and in the investment and the education about the activities in our foundation block, even though there is only 20 commercial nurseries in California, over 10,000 people are using our budwood, because, and as we saw in Northeast India, and I'm sure the rest of the India, if a homeowner can grow citrus in their backyard, they will grow citrus in their backyard. And the 10 or 20 varieties that they can find in a nursery, maybe they don't satisfy them. And as Dave said, people can go online and they say about this variety and that variety, but the local nursery doesn't have this variety. Then all of a sudden they start cutting budwood from their neighbor or from a trip that they went, and they bring budwood into California, and that's a big, big no-no for us. So we open our program, even though we are an industry-oriented program, to everybody, and you see tens of thousands of people distributing, getting material from us for home use. The insert slide down there in the corner, the blue slide, shows the first HLB tree in California. This tree had 23 different grafts on it. Someone in the neighborhood was moving around propagating citrus without knowing the risk of diseases. And that day in 2012, we start opening our system, and as you see, tens of thousands of people in our system. Everything I'm talking about in terms of quarantine for variety introduction and in terms of distribution, available to all of you. If any of you, I understand there is rules like any other country in India, and you have to have your permits and approvals and probably a project with us, but just to play around to know what's available to you, any of you can click on those two links and start the process to get material or introduce material. I will contact the national program, because as we said, they are responsible for distributing and introducing varieties for other countries or from other countries at the national level, and then we can figure out how we can execute your request. I think I'm past my time. I'm going to show you only 2 slides about, yes, we do all this in our programs, but unless if we translate it to a product and to a service for the industry, has almost no value or impact. So this is some of us visiting the Florida nurseries in 2006, when we knew that the HLB was coming in California, and we saw how well protected the nurseries were in Brazil. And when we came back to California, we educated our growers, we educated the state of California, and the state of California passed a law in 2009. All nurseries that they make commercial citrus, they have to produce them under these rules. No more outside production, they have to move indoors, they have to get budwood only from the CCPP, and so on. So by doing that, we created a big problem. We had to test over 7,000 trees, mother trees that the nurseries were using already. We had to test them within 1 or 2 years so they can be legal and stay in production. Our diagnostic protocol could test only 500 trees a year, which meant we should have informed the nurseries that they could not propagate a tree for over 10 years, which of course was unimaginable. So we perform a lot of research about high throughput processing of samples. We test the RNA quality and so on. We published our results, like exactly how you're doing in this institution. And we achieve what we call technology transfer and approval of our methods from the state. So all the hard work we did to transition the nurseries to the clean program was approved by the state. And this is my last slide. and as I say, our pride and joy that shows how clean stock programs, collaboration with the industry and the regulators and research can achieve a measurable impact. Before 2009, when the program was voluntary, if you want, you join. And we had the capacity to test only 500 samples a year, which are the bars, the red bars to the axis on the right. The black line shows that there was always a viroid infection in the nurseries. After 2009, in our high throughput sample processing research, we could test thousands of samples every year. And you see, as of 2021, 0 finds in thousands and thousands of mother trees used by the nurseries, no more viroids. We have completely cleansed the viroid, the nursery system, the nursery production pipeline from graft transmissible viroids, which is the biggest problem for us. And this final slide shows you that yes, you can build a beautiful clean stock program, but you have to make sure it's a systems approach, right? Monitor your groves, do sustainability research like the concepts of circular economy we're investigating now in our program. And thank you for your attention. I hope I didn't take too much of your time. Come for questions?
Thank you so much, sir, for your presentation and for the details, how much of planning and efforts will go into producing a clean planting program. So I once again thank all the speakers for their beautiful presentations and for their insights in the citrus research and way forward. So now I request audience to ask any questions you have regarding the presentations. Those who are present online, you can post your questions in the chat box. If time permits, we will take those questions also. So now the house is open for questions. Hello.
Sir, I'll start my questions from the last lecture to Dr. George. Sir, this clean stock program, what you have given a very excellent presentation through elaborative slides and all. We are already doing the clean stock program. Since in 1994, my first paper published cleaning of our citrus pathogens through in vitro shooting grafting. So far we have released around 5 lakhs of the planting material. They have gone in the last 10-15 years through in vitro shooting grafting. So around in 1,000 healthy orchards we have already established. So what you suggest us like to do, since there is a demand of around 2 crores of the planting stock every year in India. Through our center, we are not even meeting 1% of the requirement. And there is a great demand for the planting matter which is produced through shooting grafting. So how? So what are the other future way forward you are going to suggest us? It is already in the operation. We are already doing it. And clean stock program, it is just, I think, introduced in the recently. So what you further, where are the steps we have to intensify? So we have, sir.
One line question, what she is asking, that you have any suggestions for our conditions, very specific question. Yes.
And
For our country.
sir, some more information I want to supplement. In order to build up the more and more clean stock distribution to the growers, we have signed more than 10 consultancies, sir, for transfer of our shooting grafting based citrus multiplication, planting method multiplication through to several even. Just now today I have come back from, you say what is that company? Jain Irrigation Company we have transferred, which is a multinational company. They have taken our technology. Then Maharashtra Hybrid Seed Company, you must be knowing.
Yes.
So, 10 consultancy MOUs we have signed to transfer this technology for multifold increase of the planting stock. multiplication of planting stock, so that the growers will be benefited.
Yes.
So what are the ways you want to suggest in the present clean stock program, so that more and more good things will happen to the citrus industry? And this is one question.
He has taken a note of his question. Please take your seat.
Yes, and I'm informed about these partnerships with the private sector, and we're looking the website of the nursery the other day. I think you're doing everything that you can right now. So the question, to summarize the question is, the demand is very high for the clean stock program, but the institution and the pipeline right now cannot meet the demand, right? That's so what can you do? What you're doing is the right thing. Partnerships with the nursery industry, very, very important. We had the idea of what we were calling sentinel foundation blocks. So if we were alone in California, our program alone could not have met the demand either. So we produced the original, I guess we can call it in some cases nuclear stock, and then exactly like you have agreements with nurseries that they understand the technology and they're good, they amplifying the material for the rest of the industry. So that's, I think you should continue doing that, and that's the way, because we, the scientists, were not very good at scaling up. We're very good at the detail under the microscope and with the molecular biology, make sure everything is right. But to build the volume required, definitely partnerships with the private sector is the way to go. And you're doing it already, you just keep building on this. Yeah. The idea of the Sentinel Foundation didn't work in California for different reasons. But in addition to individual, that's what the final comment on the idea of Sentinel, in addition to individual businesses, if they can together pool resources, and let's say 5 nurseries, they have access to this foundation facility locally, and these 5 nurseries, together they manage and they contribute, that's another way. Instead of going one by one, you bring together nurseries that can work together and they have their own mini foundation facility.
That's great. Thank you so much, Gerson. And is there anyone, I understand many people, it is in hundreds, those who have attended this program by online mode, to a Zoom meeting as well as YouTube.
I have one more question.
Anyone who has joined in this program, this international webinar by online, any questions from their side? Very quick questions. Please be brief while asking your question, or you can send your question in the chat box.
Sir, online question is that which rootstock, any rootstock is showing any resistance to the HLB as of now?
The short answer is there is no rootstock showing resistance. There are rootstocks that are showing improved performance of infected trees, and this can result from a number of different things, better adaptation to the soil and so on. But there are clearly differences in rootstock performance, and our breeding program in Florida is very actively pursuing this to try and identify rootstocks that can be used by growers to keep them in a profitable condition. But no resistance so far.
Good. Thank you.
Yeah, I have one question.
Thank you, sir.
Yeah, I have one question for Fred. In one of your slide, you have shown that you are using metabolomics for identification. So there are many theories. Some are suggesting that it is the plant hormone, okay, like auxin cytokinin pathway. Some was telling it is the aspartate glutamate pathway. So actually, for my knowledge, I am asking that what are the metabolites you are targeting for identifying HLB tolerant plants or rootstock? What are the particular metabolites, whether it is aspartate glutamate or it is auxin cytokinin or any other amino acids? Even some have suggested that sugar in roots also can be a biomarker for HLB tolerance.
I can't tell you specific compounds right here and now because I'm not a metabolomics expert. I collaborate with metabolomics experts. What I will tell you is that the response of the host to this pathogen is incredibly complex, and anyone who tells you that there is one mechanism that works against this disease, or is the one mechanism involved in causing this disease is not giving you the full picture. We have seen many different things. There are metabolic differences between tolerant and sensitive types. We can look at differences, even anatomic differences. We've noticed, for example, that lemons, which we consider tolerant, they get hit by the disease hard, but they come back the following year. Our first variety, the sugar bell that I showed at the very end, is very tolerant. Also displays symptoms in the winter, comes back strong the next year. There's actually phloem regeneration that takes place. So it's like having a bypass, you know, your heart doesn't work.
Recording in progress.
The highway is blocked, so build a new highway around it. Things like this. We recently published a paper where we looked at a sweet orange mutant that's very, very tolerant to the disease, and we found, using genome sequence comparisons, that there was a large translocation that took place, and at the boundary of where this translocation took place, there's a cluster of heat shock proteins. Now, they're involved in all kinds of stress, but this particular cluster is very highly upregulated in our tolerant type. And our hypothesis, yet to be proven, is that perhaps this overexpression of heat shock proteins is able to result in protein homeostasis within the phloem tissue, leading to tolerance. So there's a number of different mechanisms. It's not so simple as one metabolite or one class of metabolites. We see differences in metabolism, in protein, in any way you look at the disease and its interaction, you'll find differences. There's just no one answer.
Fred, I have one question. Like you mentioned about the speciation in the citrus. Yeah, the species, because of the Gondwana land concept, the plate tectonics, you mentioned in that. Yeah. So did you find any co-evaluation in the literature? When did you come up with those paper? Co-evaluation with the insects among those species?
One of our team members is a real fan of the idea of this speciation resulting from changing environments and so on. And he did a lot of searching in the literature. And there are many, many examples of radiative speciation taking place at the same time in the same place, in different plants, in fungi, even in mammals. So what took place at that point in time was a huge explosion of activity across all biological spheres.
Thank you. David, one question for you. Your presentation, you mentioned the exhaustive information about the citrus cultivar. Whether this information can be aligned with the Global Biodiversity Information Facility, GBIF?
That's a very good question, particularly for India, which has really as a center of diversity for citrus and has very different economic. You're still a developing nation. On one hand, you've got large commercial producers who would very much benefit from having advanced cultivars, elite material that's been developed around the world. On the other hand, there's areas of the Northeast where they grow traditional cultivars that haven't been developed, that have not been succeeded by modern cultivars, and perhaps that's in their best interest. In terms of the preservation of traditional citrus germplasm, really that's a separate field at this point. We need to work on two tracks. Preserve citrus germplasm, primarily in citrus germplasm repositories and in the wild. And yes, we can encourage growers, say in Northeast India, to produce certain quantities of it, but it's not realistic to expect them to grow large quantities of species or cultivars for which there's no economic demand. In time, I'm afraid that they will be replaced by economically successful cultivars. The purpose of the Modern Citrus Cultivars Descriptive Database is to describe new. That modern means new. I don't know if I've answered your question, but it gives you some sense of the context.
I have one question for Georgios. I just wanted to know how useful is the thermal therapy
whether it is a supplementary or complementary to the shoot tip grafting technique. Is it really useful for the elimination of pathogens?
Thermotherapy was, and maybe in some cases still is, a very useful tool, not in our hands and not for the rhythm that our program needs to move. We cannot take our time and do thermotherapy. Plus, the viroids are not eliminated with thermotherapy. So at the end of your question, you gave the answer. Yes, we use it as a supplemental, meaning when we culture the budwood in vitro, the original budwood is cultivated in elevated temperatures, 32 or more, if I remember well, Celsius. So we give an opportunity to the young flash to escape the virus. The high temperatures suppress the virus replication. So when we harvest that tip, the chances are that it doesn't have anything in it. So we, like de facto, space, equipment, time, we eliminated thermotherapy from our activities. We're focused on STG, but we are using high temperature at the first part to help us.
Thank you, Judge. I think Prashant has noted a couple of questions from the participants who have joined online. So Prashant, please read the questions, please.
Sir, one chat, they are asking whether any microbiome related resistant towards HLB. Anything found?
I'm not personally working in the microbiome. There are people who are. Dr. Nian Wang from our research center has done a good bit of work looking at the microbiome. Myself, when I look at the work, it's obvious you can see differences in the proportion of different species that are present or absent and the changes. I find myself asking whether that is the cause of better performance, or the better performance of the tree is the cause of differences in the microbiome. I don't think anyone has yet developed or identified one specific microbe that can work.
Similar answer from work at UC Riverside. We don't know if it's a cause or effect, because a healthy tree can recruit better and so on, while a declining tree has declining populations. Some bacterial species in the rhizosphere appear to be in the category that they produce antimicrobials, and maybe they play a role. I remember if it was strepto.
I don't remember.
Yeah. But no clear path, definitely studied, unclear pictures so far if it's the cause or the effect.
So question from myself, sir. This is regarding genomics of citrus. So now a lot of genomes are being sequenced and resequenced. So whenever you are mapping a resequenced genomes, different people are using different, some are using clementine as a mapping reference, some are using sweet orange as a mapping reference. So whether this difference of mapping will affect the results, suppose you are showing that PCA, that grouping of the things, whether that different, if you are mapping to a different genome, whether that will change the results of that PCA and admixture?
Not really change the results of the PCA, because that's something different. If you're doing transcriptomics, for example, however, it will make a very large difference. Right now, I have a PhD student who's doing a comparative transcriptomic study between something tolerant and something very sensitive, very early upstream in the infection process. And he used the old sweet orange genome, not knowing that our group actually had a new phased, much higher quality genome. And he did all his work, and I looked at it and I said, that's good. Which genome did you use as your reference? He said, this one. I said, well, I'm sorry. You have to redo your project. And the results were substantially different. Globally, there's a lot of new work going on in developing new, very high quality reference genomes. And our clementine from 2011 at the time was the best, and it lasted for a long time as the most useful. Early on, we knew there were problems with it. We knew there were pieces missing. It was a haploid that we assembled. So all sorts of problems. But things are getting better and better in terms of the references that are available. Now they're phased. So not only can you look at differentially expressed genes, but you can look at differentially expressed alleles. You can look at allele-specific expression differences which adds a whole new layer of complexity, but gets you a lot more information on the biology that you're trying to study.
Since, sir, Gameter, sir, do you remember? I am Dr. Vijay Kumari. In 2006 and 7, I have written couple of letters to you. You have accepted me for working in your lab in citrus polyploidy breeding. Dr. Vijay Kumari, if you check your email,
Yes, okay.
you have invited me when I was working for, trying for the DBT overseas fellowship.
Okay.
Huh. So, sir, I succeeded in production of the citrus triploids through endosperm culture. Both in sweet orange and even Nagpur mandarin also. Even I have succeeded in production of citrus tetraploids. Sir, how far they will address? I have read you have done an excellent work in citrus tetraploid production, polyploid breeding. How can they address addressing the HLB, this problem, greening disease?
Well, I could have paid you to ask that question, actually. So we've had the situation where we've had multiple years of thousands of seedling trees in the field from our breeding program, and we've watched thousands and thousands decline and die. And we've been able to collect information over many seasons and gradually find a small subset of individuals that seem to be extremely tolerant and persistent. If we look, you know, we've made a lot of triploids. We've done a lot of triploid breeding. Tetraploid onto diploid, embryo rescue produced the triploids. If we look at the percentage of trees that are more tolerant of HLB, there are far more triploids than you would expect than diploids. So triploidy in and of itself seems to bring some greater degree of tolerance to HLB. Now, there are triploids that are disasters. I'm not saying that you have triploids, you have resistance or tolerance. No, that's not the case. But there's a greater percentage in the triploid populations that are much more tolerant than what we see in the diploids.
I agree with you, sir. What about tetraploids? Because I have studied in one of your papers in advanced citriculture systems like high density seeding and all. Tetraploids, they are conferring some resistance even to HLB. Tetraploid using as a rootstock.
Yes, many of the rootstocks that we've developed and are selecting are in fact tetraploids. But there also are a large number of diploids as well. And so the way we're doing rootstock screening for the past several years, we make populations of seedlings. We make crosses, diploids or tetraploids. We plant the seed in very poor soil, high pH, high calcareous soil, inoculated with 2 different species of Phytophthora. We put the seeds in those. Most of them don't germinate. Some germinate and die quickly, but there are some that germinate and they grow and they stay dark green in spite of these horrible conditions. The next thing we do, we will go to the field and from a tree with high titer of Liberibacter, usually a sweet orange, we'll cut a stick and we'll graft that stick onto it, onto the rootstock candidate. Cut the top of the rootstock, root it so we preserve the genotype. Take that, grow it in the greenhouse, 70–80% of them go down. We throw them away. 20% look good. We take them to the field. We plant them in a field with very high inoculum pressure, and we watch them over several years, and we begin to select. We've been doing this with both tetraploids and with diploids, and we find some very, very good rootstocks with both. The photo I showed of myself standing next to a tree that I said was a Shaquasha hybrid, that was a diploid. But we have Shaquasha somatic hybrids as well that also are very tolerant. So ploidy is a part of it. It's not all of it. It's really the genetic combination ultimately that makes the success.
Okay, sir. It will make a pivotal role in addressing this problem to some extent.
Yes.
Sir, one more question, sir. You have, in one of the papers, you have even mentioned CRISPR-Cas9 in dealing the citrus greening disease. I think you have made some success.
I haven't made success
but there was a paper
in greening resistance. There's a lot of work being done again at our station, at our research center by Dr. Nien Wang. He's had a lot of the headlines and really has a large program. I'm working with some other people who have edited some other genes. The challenge is, again, going back to the question that the gentleman asked previously about, what metabolite do you target? And I talked about how complex this disease is. Everyone has a laundry list of genes that are potential targets. And up until now, no one has really demonstrated that the single gene that they edited has been sufficient to make the plants resistant. So it's a long way to go. And most of the work has been done with juvenile tissue. So the plants are actually transgenic.
Yes.
CRISPR edits.
Yes.
Dr. Wang has developed or modified some technology to do non-transgenic editing. It's a very inefficient transformation process. The trees still are going to be juvenile. We need many years to grow them out to see how they perform in the field. We need many years to see, do they actually bear fruit? Are the fruit good? Are the trees productive? It ultimately becomes a horticultural field trial, just like whatever other breeding technology you bring to the table. So no fast answers.
Yes, sir. Then you should be ready with the efficient regeneration protocols, perfect sequencing of all the citruses, different species, to go ahead with what I feel. Madam, one question for you.
I'm sorry you couldn't come to my lab.
Yes, sir. So many administrative problems in Indian system. Otherwise, I was very happy. Thank you very much. You agreed, accepted me for working in your lab.
So thank you very much. We are running out of time, and in fact, let me announce you that Professor Fred and Dr. Georgios and Madam Chandrika and David, tomorrow also they are here.
Okay.
So we'll discuss some more questions, and tomorrow, maybe after lunch, we'll have separate discussions. But today we have some other program immediately after this program. So now I request Dr. Das for the formal vote of thanks.
Good evening. I consider it a great privilege to propose the formal vote of thanks to all the dignitaries and delegates on and off the dais, of course. Many of them are joining online also. Today's international webinar. So on behalf of all of us, I would first like to express our deep sense of gratitude to one and only Dr. Fred Gemeter, Professor of Citrus Breeding and Genetics, CREC, Florida, an eminent citrus expert, for his august presence on this occasion. Just to recall, Fred, during the year 2012, I had the opportunity to visit CREC and work with you. During that short stint, I could learn many things, especially on molecular markers and molecular diagnostics. At CREC, Dr. Fred used to be my mentor, friend, and always available and supportive. Whatever problem I used to face, I used to run to him only. So thank you, Fred. It's indeed a special privilege and great pleasure for all of us. Thus, finally, today you make it, and you are here. And I'm sure with your guidance, it will help us and rather take us to the next level for the betterment of our Indian citrus industry. So thanks. I take this opportunity to express our sincere thanks to Dr. David Karp, the citrus detective, I would say. You just saw his presentations, how nicely and how meticulously he used to document all those varieties. So thank you, David, for your nice lecture. We extend our thanks to Dr. Georgios Vitalakis, the nodal person behind CCPP. When you started our work in citrus, we used to refer CCPP as our mentor. I mean, everything, it's a model for any nursery program. So thank you, Georgios, for coming over here and sharing all of your experiences. We would also like to acknowledge our gratitude and appreciation to Dr. Chandrika Madam, last month, I think, in the citrus conference, she came, and she also took some time out of her busy schedule to come over here again and delivered our wonderful presentation. I would also like to acknowledge our sincere thanks and gratitude to our director, Dr. Dilip Ghosh sir, who has planned and chalked out this webinar within a very short time. Thank you, sir, for your valuable time and constant support. I convey thanks to all my scientist colleagues, the research associates, the SRFs, the young professionals, and other staff to attend this important program. And I also like to thank 200 plus participants from not only from this, from our country, India, 6 different countries, participants, 200 plus, they are also participating, they are online. So thanks to each and every one of them. I also like to thank our audiovisual team. Mr. Pradeep, Mr. Banerjee is here, Sunil Kumar for making available this hall, and all the other persons. I may miss somebody's name. So thank you one and all.